Today we'll do about the production of haploids or haploid culture. Okay, so if we want to create haploid plants, we have to take the tissues which are haploid. So what are they? We could take the anther, or we could take the ovule, or we could take the pollen, and we can culture that and grow them into haploid plants. Right. So let's see what's written here. There are two approaches for the production of haploid plants. Okay, so in vivo approach and in vitro approach. Okay. So let us look. Okay. Haploid plants, they possess only a single set of chromosomes. Okay, one complete set, but they don't have them in pairs. They have just one set. That is, the number of chromosomes is the gametophytic number, which is N. Okay. I think if I zoom it's better, isn't it? I think so. Diploids, they contain two sets of chromosomes. And haploid plants, they are important because we can produce haploid lines or homozygous plants for the improvement during plant breeding programs. Okay, sometimes we want to breed a better, higher quality, better, higher hybrid. Okay, so we use haploid culture. Now, haploids were discovered by Berdner in Datura stramonium. Okay, so it was first discovered in the Datura plant. So plant breeding experiments have been done to develop haploids. Indian scientists Guha, okay, this is not Kuha, it's Guha, G-U-H-A, so you please correct it. Guha and Maheshwari, they showed that haploid embryos can be produced from plantlets, from microspores of Datura inoxia, okay. And then other scientists, okay, you can read on your own. Then let's see how we can group our haploids. We can divide them into some categories: monoploids or monohaploids and polyhaploids. But what are monoploids and polyploids? Okay, maybe this is better for you. Okay, monoploids are haploid that possess half the number of chromosomes from a diploid species. Okay, and then polyhaploids, they possess half the number of chromosome from a polyploid species. Okay, let's say monoploid means first we have a diploid, right, 2n, half of diploid will be n, then polyploid will be a polyploid plant. No, let's say you have 4n, half of 4n is what? 2n. Half of 6n is what? 3n. Okay. Half of 8n is what? 4n. Okay. So these are your polyhaploids, and this one in front is your haploid. Okay, or monohaploid. So I think it's clear from there. So in vivo and in vitro approaches, do you know what is parthenogenesis? Like without any fertilization, no? Like from the egg only, a zygote formed by itself. Yeah, parthenogenesis is just like Jesus, no? He came from the womb of Mary without any fertilization. So that is when you produce spontaneous haploids. Okay, so just imagine that Jesus was as a haploid individual, because he was formed directly from the egg or the ovum of Mary. It's just it's just an example. Okay, it doesn't mean you write that in your exam, just an example. So when I say parthenogenesis, it means directly a haploid plant came from a haploid set of chromosomes. Okay, there was no fertilization, and it occurs in nature sometimes via parthenogenesis. Okay, now let's look at in vivo techniques for haploid production. So for what I was talking about, parthenogenesis and apomixis, when haploids come in spontaneously in nature, this is your in vivo. Okay, in vivo means in nature. This one in vivo. Okay, then if we look at the in vitro approaches, means in the lab. Oh, sorry, this is in vivo and say in vivo, in vitro. Yes, okay, there's many. Okay, let's look first at the in vivo techniques for haploid production. The first one is androgenesis, then gynogenesis, then distant hybridization and irradiation effects, chemical treatment. Okay, so there are 5 in vivo techniques to produce haploids. So if we start with the first one, androgenesis, what it means: development of an egg cell containing a male nucleus to a haploid is referred to as androgenesis. Okay, so it make no sense, no? Okay, let's say this is the egg cell. Okay, this is the egg nucleus. So what you do is you remove that egg nucleus, you kill it somehow, and then you just introduce the sperm cell. Okay, and then it will release its DNA there, and this when it has released, the sperm has released the DNA, that DNA is the DNA of the male, is it not? Then from this egg, okay, you will develop a haploid plant. I think it's clear. That's why egg cell development that has a male nucleus. Okay, so I think it's clear. So now the next one, gynogenesis. Then gynogenesis, simply you don't fertilize the egg, but you just manipulate it so that it will develop into a haploid plant. Okay, so you just culture it. Then distant hybridization, you will just eliminate one of the parental genomes as a result of distant hybridization. Okay, let's say when you hybridize plants that are so unrelated to one another, one of the parents' genome does not succeed. Okay, so simply one parent genome will just be inactive, and the other parent genome will develop into a new plant. Then irradiation effects. You can use ultraviolet rays or X-rays to induce chromosomes to break, and we eliminate those chromosomes and will produce haploids. So let's say this is a single set, this is another set. So you use X-rays or ultraviolet rays, and then this will be destroyed, that chromosome, and then from here only, since only N is left, then it will generate into haploid plants. Okay, but it doesn't mean it's very easy. In lab it's very tough to do this, but I'm telling you the techniques. Okay, it's very tough to do. Chemical treatment, we can treat with certain chemicals like colchicine, chloramphenicol, nitrous oxide, malic hydrazide, etc. The most common one is this one, colchicine, and these chemicals, they induce chromosome elimination. Okay, this colchicine, no, suppose you have, this is pair, no, this colchicine is such a chemical that it attacks extra pairs. All, like it's a type of chemical, no, that attacks and binds to one of the pair, and only the single set will remain. Okay, so these were your in vivo techniques. What were they? Androgenesis, You just the sperm, okay, remove the egg nucleus, gynogenesis, just the egg, you grow it. Then distant hybridization, distantly related crops, one parent will not be functional, and then you irradiate with ultraviolet or X-rays to break the chromosome, and then you treat with chemical like colchicine to remove the pairs, okay. Then the in vitro technique, this is there are two methods for this, androgenesis and gynogenesis, okay. In vitro means in the lab, okay. So we can do androgenesis, we can produce haploid plants by anther or pollen culture, and these plants will be androgenic haploid plants, okay. Then gynogenesis, ovary or ovule culture, and we produce haploid plants known as gynogenic haploids, okay.